นุษย์เราเริ่มตั้งคำถามเกี่ยวกับกลไกการทำงานพื้นฐานของธรรมชาติมาตั้งแต่ครั้งโบราณกาลองค์ความรู้ที่ได้ถูกส่งต่อจากรุ่นสู่รุ่นถูกพัฒนาปรับเปลี่ยนต่อยอดผ่านการค้นคว้าทดลองตั้งสมมติฐานและพิสูจน์ทางทฤษฎีเพื่อให้เข้าใจถูกต้องมากขึ้นฟิสิกส์ทฤษฎีถือเป็นวิทยาศาสตร์พื้นฐานที่สำคัญอันหนึ่งที่อาศัยแบบจำลองทางคณิตศาสตร์ในการไขความลับความเป็นไปของสิ่งที่อยู่รอบตัวเราตั้งแต่ระดับอะตอมไปจนถึงระดับจักรวาลส่วนหนึ่งของผลสำฤทธิ์ที่มนุษย์ทำความเข้าใจธรรมชาติผ่านภาษาทางคณิตศาสตร์คือเทคโนโลยีอันหลากหลายที่อำนวยความสะดวกในชีวิตประจำวันไม่ว่าจะเป็นโทรศัพท์มือถือคอมพิวเตอร์หรือแม้แต่โครงข่ายอินเทอร์เน็ตที่ทำให้โลกอันกว้างใหญ่แคบลงไปเพียงปลายนิ้วสัมผัสยังมีคำถามอีกมากมายที่เรายังตอบไม่ได้ทั้งหมดเกี่ยวกับธรรมชาติซึ่งถือว่าเป็นเรื่องที่ท้าทายและรอคอยให้นักวิทยาศาสตร์เข้าไปค้นหาคำตอบในอนาคตในปัจจุบันบุคลากรของวิทยาลัยเพื่อการค้นคว้าระดับรากฐานถือได้ว่าเป็นส่วนหนึ่งในการค้นหาคำตอบของคำถามข้างต้นผ่านการทำงานวิจัยหลากหลายที่ให้หัวข้อที่ล้ำหน้าวิทยาลัยเพื่อการค้นคว้าระดับรากฐานสถาบันสำนักเรียนท่าโพประกาศจัดตั้งขึ้นเมื่อวันที่ 14 มีนาคม พุทธศักราช 2554 รับรองสถานะโดยสภามหาวิทยาลัยนเรศวรให้สถาบันสำนักเรียนท่าโพสำหรับฟิสิกส์ทฤษฎีและจักรวาลวิทยาหรือ TPTP ซึ่งแต่เดิมนั้นดำเนินการภายใต้จิตอาสาของเหล่าคณาจารย์ฟิสิกส์มาสเตอร์ทัพพาเอียนตั้งแต่แรกเริ่มเมื่อวันที่ 8 สิงหาคม พุทธศักราช 2539 ต่อเนื่องยาวนานจนเป็นสถาบันแห่งการผลิตผลงานศึกษาวิจัยและเป็นอาสมบ่มเพาะนิสิตสาขาวิชาฟิสิกส์ทฤษฎีให้มีความรู้ความเข้มแข็งทางวิชาการจนได้รับการยกสถานะให้เป็นหน่วยงานระดับคณะวิชาอย่างเป็นทางการถือเป็นการประกาศเจตนารมณ์ผลักดันยุทธศาสตร์พิเศษของมหาวิทยาลัยนเรศวรที่จะก้าวเป็นมหาวิทยาลัยวิจัยชั้นนำของประเทศในระยะเวลา 10 ปีที่ผ่านมาวิทยาลัยเพื่อการค้นคว้าระดับรากฐานได้ผลิตผลงานต่างๆมากมายผลงานวิจัยตีพิมพ์ระดับนานาชาติจำนวน125บทความผลงานวิจัยตีพิมพ์ระดับชาติจำนวน8บทความงานประชุมวิชาการระดับนานาชาติจำนวน7ครั้งงานประชุมวิชาการระดับชาติจำนวน10ครั้งการจัดอบรมเชิงวิชาการระยะสั้นและโครงการบริการวิชาการจำนวน58โครงการการจัดสัมมนาวิชาการ200ครั้งคณาจารย์ของวิทยาลัยได้รับรางวัลเชิดชูเกียรติทางวิชาการจำนวน15รางวัลบันทึกข้อตกลงความร่วมมือกับสถาบันชั้นนำอื่นๆจำนวน13สถาบันหนังสือและตำราวิชาการจำนวน13เล่มวิดีโอสอนออนไลน์ผ่าน Asst Channel จำนวน34วิดีโอนอกจากนี้ Asst Consortium ได้สร้างเครือข่ายความร่วมมือทางวิชาการระหว่างสถาบันฟิสิกส์กว่า20สถาบันกลุ่มเครือข่ายพิษณุโลกฟิสิกส์เน็ตเวิร์กได้ส่งเสริมกิจกรรมสัมมนาวิชาการในจังหวัดพิษณุโลกและสถานีฟิสิกส์ศึกษาคือการให้ความสำคัญกับครูผู้สอนฟิสิกส์ในโรงเรียนระดับมัธยมศึกษาให้เข้มแข็งและถ่ายทอดสู่นักเรียนผู้เป็นอนาคตของประเทศที่ผ่านมาวิทยาลัยเพื่อการค้นคว้าระดับรากฐานได้ผลิตบัณฑิตสำเร็จการศึกษาในหลักสูตรวิทยาศาสตร์มหาบัณฑิตจำนวน8คนหลักสูตรประกาศนียบัตรบัณฑิตชั้นสูงจำนวน10คนหลักสูตรปรัชญาดุษฎีบัณฑิตจำนวน5คนผลงานเหล่านี้จะสำเร็จขึ้นมาไม่ได้หากปราศจากผู้สนับสนุนและแหล่งทุนวิจัยอาทิมหาวิทยาลัยนเรศวรสำนักงานกองทุนสนับสนุนการวิจัยสกว.สำนักงานการวิจัยแห่งชาติวช.โคร รงการปริญญาเอกกาญจนาพิเศษคปก.นิวตันฟันด์ศูนย์ความเป็นเลิศด้านฟิสิกส์เทพสถาบันวิจัยดาราศาสตร์แห่งชาติองค์การมหาชนมูลนิธิโทเรเพื่อการส่งเสริมวิทยาศาสตร์ The Abdus Salam International Center for Theoretical Physics ICTP อื่นๆปัจจุบันวิทยาลัยเพื่อการค้นคว้าระดับรากฐานมีคณาจารย์ทั้งหมด 9 ท่านโดยแบ่งเป็นคณะผู้บริหารวิทยาลัย 4 ท่าน 3 ห้องปฏิบัติการวิจัยห้องปฏิบัติการวิจัยจักรวาลวิทยาและความโน้มถ่วงห้องปฏิบัติการวิจัยทฤษฎีสนามควอนตัมและฟิสิกส์พลังงานสูงหน่วยวิจัยฟิสิกส์ดาราศาสตร์และจักรวาลวิทยาเชิงคำนวณจากอดีตถึงปัจจุบันเรายังคงดำเนินการภายใต้พันธกิจสำคัญอย่างต่อเนื่องดังประกาศเจตนารมณ์ท่าโพณวันที่ 8 สิงหาคม พุทธศักราช 2539 เราจะสร้างโรงเรียนฟิสิกส์ที่ดีที่สุดแห่งหนึ่งของประเทศขึ้นที่นี่โดยหวังเป็นอย่างยิ่งที่จะส่งเสริมสังคมแห่งการเรียนรู้จุดประกายเยาวชนและสร้างบุคลากรให้มีความเข้าใจลึกซึ้งเห็นถึงความสำคัญของวิทยาศาสตร์พื้นฐานต่อยอดวิทยาการความรู้ด้านต่างๆให้ก้าวหน้าบนรากฐานที่มั่นคงทางวิทยาศาสตร์ซึ่งเป็นกลไกสำคัญให้ประเทศไทยขับเคลื่อนไปอยู่ในระดับแนวหน้าของโลกในอนาคต
Good afternoon, everyone, and welcome to IF Colloquium Online Seminar, Series number 6, organized by the Institute for Fundamental Study, No.6 University, Thailand. My name is Kalin Yukong. I am IF Deputy Director of Strategy, and it is a pleasure to welcome everyone to this afternoon online event. Let me first give you the idea why do we need to do this kind of activity. Most of the time, academic conversation would normally circulate among scientists, yet it's bridging the gap between the science world and the general audiences is very crucial for this country in this current competitive world. Then the Institute for Fundamental Study would like to provide a platform for people who are interested in physics, mathematics, and other related fields come to enjoy the progress of the advanced research. For this talk number 6, we are very pleased to have Dr. Thana Sutthibutpong from King Mongkut University of Technology, Thonburi, to give a talk on a topic protein engineering from physics point of view. Okay, let me spend a bit more time to introduce our speaker formally. Dr. Thana Sutthibutpong completed his bachelor degree in physics from Prince Songkla University, Thailand, and then he went further for his Ph.D. in physics in University of Leeds, United Kingdom, actually where we met right there at Leeds. At the moment, he's working as a lecturer at the Department of Physics, Faculty of Science, King Mongkut University of Technology, Thonburi, Thailand. His research interest is on, molecular modeling, simulation of the biomolecules, polymers, and carbon-based material, and of course, protein engineer, that kind of stuff. And well, the talk will be roughly 40 minutes long. And before I stop doing the talk, if you have questions, please put them in the comments. Our staff will collect all the questions and deliver to the speaker at the end. Anyway, without further ado, let me give this screen to our speaker, Dr. Thana Sutthibutpong. Okay.
Okay, Kap. So thank you, Dr. Sikrin, for a very warm introduction. So my name is Thana or James. I'm from Physics KMUTT. So yeah, as Pirin has already introduced, I am interested in many sorts of molecular modeling, and protein engineering is one of the interesting fields that I have been exploring for many years. So let's start with the outlines of this presentation. I will start with introducing the audience to proteins in perspective of biology, medical, and biotechnology. And then in the next part, we are going to see protein in another perspective, which is seeing protein as a physics problem. We are going to see how and why protein folding has become such a big problem, and how humans can solve this pretty big problem. And then in part 3, it's going to be on the applications of, and one of the benefits when we have already solved the structures of proteins. So we push forward to do protein engineering, and I try to introduce you to some of the work that I have done and some ongoing ideas. Okay, so let's start with the part 1. So introducing proteins in biological world. So proteins, many might have known from biology 101 as is macromolecules comprised of folded chains of amino acids. So amino acids, we got it from eating food, and protein from meat, from beans, got digested into shorter chain, it getting shorter and shorter and shorter, and the shortest unit is called the amino acid, and the amino acid itself can reform into the polymers called proteins, and proteins can function as a building block of life, can function as muscle, cytoskeleton, and also as a catalysis tool or enzyme. So we normally use proteins to move, to do many things, and also to accelerate chemical reactions within our body. And then how can we shape the protein to what it looks like? The proteins can be shaped by and can be encoded by genes, which is said in the central dogma of molecular biology, where from the DNA, from the genetic code, DNA get transcribed and get translated from mRNA into proteins. So for proteins in medical studies, we're talking about how we are going to interrupt the pathways. So the word pathways in biomedical research, it is a series of chemical reactions that happen in cells. So proteins in this regard will be working as a signal receptor and also as a catalyst. So it's getting somewhere within this. So yeah, all the circles here, they are mostly the proteins, and you can see that the pathways for cancer. This is the pathway for cancer. There are a lot of protein involving in this process, and there are kind of series of chemical reactions that might cause the malfunctioning of the body and cancer. So in biomedical research, many times we are attempting to interrupt this signal by designing the inhibitor. So normally the idea of designing the inhibitor or the drug molecule is we just need to design it in the way that it has the stronger interactions to the proteins than the native substrate, so that it can come and compete and kick out the native substrate and stop the process. Then protein in biotechnology field. So the main idea is we are talking about how to speed up the chemical reactions. So the ultimate task for many applications of protein or enzymes in biotechnology is we want to convert waste into good. So bio-waste or the biological waste is the waste from agriculture that we get about like 1 million, 2 million tons every year, and then normally people just burn them for good, and that causes like PM 2.5 and many things. So we trying to think about alternative ways to get rid of them, and also try to convert them into something good. Such, for example, this is the bio waste from sugar cane, in which the lignocellulose complex consists of a lot of like complicated polymer represented by this diagram, and we are using some kind of enzymes as a converting machines to convert this complex into smaller molecules. So if we reduce them into the smallest molecules, it becomes xylitol, and then or maybe it's like a short chain sugars that can be the prebiotic or can be a food for gut microbiome. So can be some stuff that is more healthy. But the thing is, these kind of chemical reactions happen pretty slowly. So we can think about having like bio waste, having sugarcane waste, throw some liquid, some EM liquid into the bio waste, but you need to wait forever until it being converted into some goods. So we are trying to work hard to speed up these converting machines. So that was in the proteins in biological, in biotechnological, and medical worlds. And then now we are talking about the protein as some physics problem. So we're talking about firstly the criteria for the protein folding. So in this picture, so I'm trying to represent like the step or the criteria of protein folding, where the proteins can be structures can be classified as the primary, secondary, tertiary, and quaternary structures depends on the level of folding. So we talk about primary structure first. So primary structures, as we mentioned before, that protein is a heteropolymer, consists of amino acids of different types. So we try to think about the protein as a chain that is very, very heterogeneous. So one unit of the chain, which is amino acid, it could be the combinations of 20 types of amino acids. And each type of amino acid on the right-hand side, you can see that we have grouped them by the physical properties. Some of them, they are non-polar, they have non-polar side chains. Some of them, they have positively charged side chains, some of them negatively charged. And some of them, they are non-charged but polar, so they behave as a dipole moment. And the wonderful thing is, this thing can be encoded by DNA. So from the table on the left, the DNA code, the codon, can be translated into the chemical structures with 20 different physical characteristics on the right-hand side. So that was the primary structures. Then we come to the secondary structures when the series of amino acids get combined. So they combine, they interact, and then they fold into some shapes such as sheets, helix, and we can also plot them and try to define some kind of phase diagram of the protein structures according to the backbone torsional angle. So you can see from this torsional angle phi and psi that protein can develop into many different shapes by rotating these bonds, and some pattern of shapes, some combinations of these torsional angles. You got alpha helix that look like this in 3D. Sometimes you got beta sheet that look like this in 3D. But many of the time, you would just end up with random quality of polymer. Okay, so this is the example of the folded proteins. So this combination of amino acid sequence, in which we have some of them hydrophobic and we have some of them positively charged. It could form the alpha helix, depending on the interactions between backbones and between side chains. And then we're talking about the classical tools to predict how the short peptide, how the short chains of protein can fold. So this tool, it has been implemented in the server called BepFold. So in BepFold, they start off with using a coarse grain representation. So the representation on the left-hand side, we are trying to represent the structures of proteins by the all-atom representation. So to compensate the computational cost, they try to convert these all-atom representations into just one coarse grain beads. So one approximated beads to reduce the degree of freedom and the searching time for the algorithm. And then for each of the coarse grain beads, they need to optimize for the parameters for Coulomb repulsions and van der Waals interactions, and then they need to optimize them for all pairs of interaction. If I bring an amino acid lysine and I bring amino acid arginine, lysine and arginine should have their own definition for pair interactions, and it would be encoded into this parameter epsilon and Rij for interaction range and interaction strength. And then after constructing this model, they bring them into the search algorithm, and then they try to optimize the conformation or optimize the configuration of the chain to have the minimum energy. So that was the classic way to approximate the structures. And then we push forward to the tertiary structures, or even more complicated kind of folding. So in tertiary structures, by definition, is the folding of the secondary structures into some unique global shapes. So you can see like many kinds of different shapes, but all of them are made of chains of amino acids, but different combinations. And the shape, of course, is determined by the amino acid sequences. And then the fourth order, or the quaternary structures. So when we have one big piece of folded structures in tertiary structures, quaternary structure will mean to assembly the tertiary structures, to assembly the protein blocks into a complex, and that would facilitate the biological functions. So for example, if you can see the schematic diagram of the virus capsid of SARS-CoV-2, for SARS-CoV-2, if we just cut the part of the spike protein is already a quaternary structures that consists of many of protein domains assembled into a complex. Okay, and why protein folding becomes such a big problem in terms of physics, as we try to understand how protein can develop many configurations in the conformational space. If I consider each of the torsional angle, which can develop into different shapes, we can see that if I try to plot the potential energy function or landscape of just one torsion angle, so we have three energy minima here. So when we have three energy minima, presumably we have three possible configuration or microstate for each of the torsion angle. And then for one amino acid, we have two torsion angles. So that means it is like already like 9 possible configuration, approximately 9 configuration for each amino acid. But normally for one protein molecule, for one fully functional enzyme, we have the number of amino acids around 100 to 1,000. So in case that we have a protein with 300 amino acids, it could develop the shape into by the number of 10 to 286 microstates. So those are like possible shapes that a protein can develop with this size can develop. So so how to solve for the right structure? So if we come to look at the sequences, so for these sequences, it has 20 different kinds of amino acid or 20 kinds of physical properties that will become the constraint to fold the protein into shapes. But still, the different number of amino acids could also result in many different types of constraints anyway. So that is how this becomes so hard. So and then when we come to see the protein folding and misfolding energy landscape, so if we try to plot the potential energy function in some of the principal coordinates, we can see kind of very rough folding energy landscape. That means just only little perturbation, little drive can drive the proteins into some different stage. So that could cause the misfolding of the proteins. So misfolding occur when with perturbation from the environment, or sometimes occur with mutation. So if, for example, if I start with the native state, that protein can fully function in the body. As it overcomes some environmental factors such as low pH or maybe high temperature, it could climb up this hill and somehow it could end up to be amorphous aggregate, or somehow it could go very wrong and end up to be amyloid fibril. And this kind of amyloid fibril could be the cause of many diseases. So this is how possible that protein folding can go wrong in our body, and this process is happening all the time, slowly. So to solve this, to solve the protein folding almost completely, so there are people from DeepMind team that developed the code called AlphaFold. So it is a deep learning platform that trying to learn the data of protein from the protein data bank. So as you can see, the data from the protein data bank will consist of the coordinates. So coordinates of all the atoms within the proteins, and will consist the data of the protein sequence. So protein sequence is also corresponding to the DNA sequence. So we bring this data to train the model. Actually, they brought this data to train the model, and for us user, if we bring the primary structures, the primary structure that would just look like letters because they are protein sequence or amino acid sequence. From the patterns that default, DeepMind, and AlphaFold has learned, one can predict the coordinate of the proteins, and then from the tests of the AlphaFold themselves, they have found that the level of confidence is above 90% from the experiment. So just only not more than 10% error from experiment, which is very impressive. When we try to have a look through like 10 to 300 possible microstates, this is very brilliant. So the composition of this code, it consists of the sequence alignment and structural database pairing. So that is the one part that they work on the bioinformatics to pair the sequence input with the database. So that they can bring structures from many of the organisms to use as a predictor for the proteins, and then they try to, and then the part that is called structural model, the biophysics part of this, is they try to create a simplified model of proteins. So they view the backbone of the proteins that always look the same. Okay, so for the protein structures, the backbone or the main chain look the same, but the things that can be vary or different are the side chains. Okay, these are the side chains. So they try to view the backbone as a rigid body that can be rotated, that can have many different like relative orientations. So they kind of like trying to learn the pattern of relative orientation and translations, and they try to predict what can happen. So if we look from a pair correlation matrix, we can say that maybe the component that's close to each other, that would be the part that determine the alpha helix, the one that a little bit further that could determine the beta sheets, and the one that then, and the components pairs, those are further away, they can predict the folding of the secondary structure into tertiary. So they can predict like the higher order of folding. Okay, and for the benefit of knowing almost exactly how protein folds, is about designing inhibitors or drugs that we talked before, because it is impossible to design the chemical molecules to bind with some substrate, to bind with some target proteins. It's impossible if we don't know how exactly the chemical details of the proteins. So that was the AlphaFold has also offered us. So they also offer like the full chemical details of the proteins, so that we can use it for the drug designs. And then knowing exactly how protein fold could benefit protein engineering. So about engineering. So we just borrowing the words from the actual engineering of the building or of the machines. So we try to think about protein as a machine. So we replace, we alter some parts of proteins as the machine to improve the efficiency. So there's going to be some working on in the theory, in the simulation lab, and in the wet lab, in order to get the better enzyme for every working cycle. So for summary for this protein physics 101 part. So proteins is a sequence of amino acid that can be folded into shapes constrained by heterogeneous interaction fields created from different amino acids. Folding landscape can be broad and very rugged, and small perturbation could cause misfolding and could cause many things wrongs. And for full structure information can enable us to study drug design and protein engineering. So then part 3 about protein engineering, where I give you some more examples. So the idea, the motivation of protein engineering, many times, it comes from the evolution in the molecular scale. So we are now focusing on some organisms that are living in some harsh environments, such as in this hot spring in Yellowstone, USA, that they discover some bacteria. Those are extremophile, so they can live in hot water. So when we look inside, inside this bacteria, there should be some enzyme that evolve in the molecular level in order for the bacteria to survive this condition. Otherwise, they get natural selection by the process that enzymes are melted or denatured. So the keys for survival of the organisms are to possess the enzyme that can also survive. So for protein engineering, we try to simulate, try to imitate, try to make the same kind of evolution. Okay, and for the protein reverse engineering, we try to do some kind of deep analysis to understand how this can work. So a little bit about enzyme kinetics. So these are the set of differential equations that talking about the rate of enzymes that can generate the product in the red line. So the solutions for these differential equations are here, in which the reaction velocity, which is also defined by d by dt, which is pretty much the same as the velocity. It depends on the constant KMUTT, Kr and Kf. So Kf, or the forward constant, it denotes the probability for the substrate to binding with enzyme, and Kr, reverse constant, it represents the probability of the substrate to be unbound from the enzyme, and the kcat, or catalytic constant, it denotes the probability of the reaction to occur at the enzyme. So if we try in some way play with these terms, we can play with the rate that we can produce the product for the enzyme, that we can produce more product of the enzymes. So for Kf and Kr terms, they are related to Gibbs free energy by considering each term of the interactions between the substrates and the enzyme. So interactions between the substrates in green and the enzymes in gray can be broken down into electrostatic, van der Waals, and effects from solvation. So we can either be working with binding of the pocket that kind of like trying to hold the substrate very tight, or we can try to work with KCAT directly, that we try every way to bring the substrate to be more closer to the catalytic site and to have the reaction there. So as we mentioned before, engineering procedure need some collaborations between theory and experiment, because this experiment to do side direct mutagenesis to actually change the DNA of the enzymes and try to do PCR, try to purify the enzyme and try to transfer it into bacteria and let the bacteria to produce the enzymes, and we bring the enzyme to test the activity in the lab. This takes quite long, but compared to our work in that we try to do protein engineering in the computer, if we want to change the sequence, we simply open the Microsoft Word, push delete, and type the new one. That's our engineer. And so this is the way that theorists can try to help the experimentalists. So if we are having about to have better enzyme for each cycle, so we use theory or simulation results to pinpoint which one is good, which one is bad. We can reduce the number of cycles to get the better enzyme. So for numerical simulation, it is the combination between chemical data, physics, computing tools, and we get some knowledge from that. So for chemical data, we have X-ray crystallography, NMR spectroscopy, and which we get the protein structures from that. We have the PubChem database where we get the information of the chemical molecules from there, and we have the activity measurements in the lab to be our data. And then in the simulation models, they incorporate classical mechanics, electricity, statistical mechanics in order to build models and do some simulations. So for computing tools, it incorporates numerical integrations and algorithm to regulate pressure, temperature, and then we have some way to approximate the potential to be accurate enough and to be efficient enough to be used in the computer. So normally to transfer the parameters from quantum mechanical scales, we kind of like doing some kind of classical parameterization to transfer the details of electronic distribution around the molecules calculated by the first principle of quantum mechanics model, and then bring that parameters to build this kind of like ball and stick models in the computer, which are the classical molecular dynamics. So in classical molecular dynamics, we think about the protein structures from the database as initial positions in our system of particles. And then we have the file that contain all physical information. So we need to define how each atom connected by some sort of artificial string. And we need also to define the interactions that not come from covalent interactions. So which could be Coulomb repulsion, which could be van der Waals interactions. And then after that, use some numerical integration to solve a very large number of Newton's equation in the computer, and use some kind of statistical mechanics to regulate the temperature and pressure to be according to the canonical ensemble. And then as the output, we have kind of like moving trajectories, just like the x at any time t. So it was actually simply trying to solve a very large number of equations of motions. And as the system is equilibrated, we bring the equilibrated trajectories for further analysis. So for some case studies, our group try to study about the thermostability of the enzyme using this idea. So we try to create some mutants based on the knowledge on the extremophiles. So we try to read some papers about many of the extremophiles and try to do the same as the reported extremophiles. And then by doing simulations, we come across some interesting mutant that has the lowest unfolding rate in our simulation, and then when we bring that mutant to test in the lab, it turns out that that one was the only mutant that can work within the over 100 degrees Celsius in the lab. So, and we can explain that the additional disulfide bond. So we try to create additional bonds between some important parts at the back of the protein. So it could somehow reduce the conformational entropy, and it worked the same as as we use sulfur to vulcanize the rubber. So that was one of our discover mutant. And we try to further study on the same enzyme by analyzing the pathway of the unfolding, and we try to, and then we have located some weak spot for the mutation, for further mutation. But the thing is, we have a tilting problem, that the newly created mutant that was designed, they decrease the activity. So the activities come down to merely 1,500 from our 3,000. But so so we try to look closely at the structures, and we found that mutation at that point created some tilting or the distortion of the protein shape, and when we try to analyze the interactions between substrate and the proteins, so we see the profile that doesn't look balanced. So because of the angle doesn't look right. So we try to create another mutant that used the same old mutant from the last study, and then we kind of can solve this tilting problem. So we mutated in one way that we think that was good, but it could distort the structures, and then we try to repair it in the other way, and yeah, it becomes all right. Pretty hard to understand. I still don't really understand it thoroughly, but as we see it from the simulations, we sort of have a little bit belief on it. So and also when we taste it in the lab, so it turns out that this new mutant that we try to synchronize between two mutations, it actually increasing the activities. But there is another turnaround for the K40L that we thought it was a fail. But when we try to analyze the product very closely, we found that K40L mutant, it cannot catalyze, or it cannot break down the tetramer. Okay, so if we talk about polymer, of this polymer, we can think that X1 is the smallest unit, X2 is the dimer, X2 trimer, X4 tetramer. But K40L, from K40L, we further analyzed the profile, and we found that the enzyme can digest the polymer up to X4, and then the rate becomes lower. So and then it became somehow good, because X4 is actually the food for that is favored by some of the bacteria, some of the gut microbiome, and it turns out to be the high value byproduct. So it's pretty interesting to have a further study, or the microbiologists could work on it to isolate this high value product, and to make it real. So as I have mentioned, there are still many points that I really don't understand thoroughly from the simulation. This just happens in the simulation. That just happens in the simulation, and I don't understand. So I make the conclusions about the atomistic MD that I use it as a tool to visualize the behavior of single representative molecules, but to understand better the local effect of local change to global behavior in both evolution or in protein engineering, we might need some further methods. So then last year I turned to study more with my student with top about the network topology. So there is a paper from Estrada. So he is a very famous guy in information theory. So published many publications and many books. and he tries to create a new way to represent the protein. So by simplifying the proteins into the network that connect the protein residues. So in this representation, Estrada just brought one atom from one amino acid residue to be as a node in the network, and try to create the edge of the network by considering the distances. So here just only a rough approximation that the network is formed when the distance is closer than 7 angstrom. But yeah, here this can be changed if we try to do some coding. We can try to change this. So from constructing the network, we come up with the matrix called adjacency matrix. So that is the way that we represent this node and edge stuff, and we try to calculate some parameters, some measurements of the network. So there are the first kinds of measures for parameters such as edge density, clustering coefficient, average path length. So they are the parameters that relying on the nearest neighbor only. And then there is a second category of measure. So from the adjacency matrix A, we try to create another matrix called G, in which G is the exponential of the matrix A. So having this exponential to be in a Taylor expansion, so we can write this G matrix as a power series, and we have the A power k. So we can think about A or adjacency matrix as a walking operator. So if we have A to k, it is a k-step walking operation. So which means it is the transferring of the signal between nodes, between one node to another node, or between one node and back to itself, by using k-step, and we try to see how many possible microstates it could have. So that we construct the metric G and try to calculate some terms in network topology, such as subgraph centrality and communicability. Okay. And we got some case studies that we trying to reproduce Estrada by let's say that it could be a reverse engineering of SARS-CoV-1 and SARS-CoV-2, how they evolve from SARS-CoV-1 and SARS-CoV-2, and how SARS-CoV-2 is doing better in terms of network topology and communication of the mechanical signals along the structures. So from Estrada, and they have found that for SARS-CoV-2, somehow they found that the average subgraph centrality and average communicability become significantly higher than the SARS-CoV-1. And then when we try to bring the trace of the G matrix or the subgraph, all the subgraph centrality to plot, they found that at the peak that has the highest centrality, it is the amino acid residue that's sitting next to the catalytic site, sitting next to where the chemical reaction occurs. And then now the work in progress from me and my student is we trying to push further the work from Estrada to look at the evolution of from SARS-CoV-1 to SARS-CoV-2 by looking at all the positions in these main protease enzymes that have the mutation, and we're trying to map the site of mutation onto the difference of the subgraph centrality between the SARS-CoV-1 and SARS-CoV-2. and we found, and we could say that we might find some correlation, but not 100% sure yet. But when we try to map in three-dimensional the positions that those are mutated in the red, it also corresponds to the positions where the communicability between nodes within SARS-CoV-2 became larger than the communicability in the SARS-CoV-1, and yeah, this need more investigation, and we're thinking about try to analyze these on the other proteins, because these main protease from SARS-CoV-1 and SARS-CoV-2, they are the enzymes, and then we might try to bring the same network calculation to analyze the profiles of the other enzymes as well. Okay, so conclusions. So I have shown that molecular modeling techniques can serve as a tool for understanding effects of site-direct mutations in the protein engineering to increase the efficiency of the enzymes. But more analysis are needed to better understand effects of local change to global behavior. And then we have tried to reproduce the work from Estrada, that we use network topology to analyze the effects of mutation from SARS-CoV-1 to SARS-CoV-2, especially in the main protease enzymes. So my last sentence is, I think solving protein structures by AI is brilliant, but it's still not the end of the story. So there are still more open questions on the disruption of structural networks and dynamics of proteins during evolution and protein engineering to be investigated. Okay, so that's the end of my talk. Thank you.
All right. Okay. Well, thank you very much. Oh, with this kind of very, very interesting talk, I would say. Yeah. Yeah. So I believe that this kind of study or this kind of research would benefit a lot to the humanity, right? Because, well, if I understand correctly, because you try, well, it's going to be something to do with the drug design as well, right?
Yeah.
And you did try to see the well, about where the original, the problem of the, like a disease like cancer, actually from the molecule structure, right? Something like that. It would, well, definitely benefit us. Yeah.
So it is not normally is not originally from the idea of our group, but we try to behave as, you know, I always like tell my students that we can behave as a tool for many people, as a tool for microbiologists, for maybe bioinformaticians, maybe from chemists, that they if they want to understand further the problem, we can try to do some analysis for you. Yeah.
Okay. Good. Anyway, so for the online audience, if you have questions, please, well, you can put in the comment. I will bring to the speaker. But, well, for at the moment, you talk about landscape right here. Well, I believe that in physics we have, well, many kind of landscape, like in different subject, like in string theory we have like a string landscape. Yeah.
Oh.
Right. And then you also have the landscape as well. Right. But I believe is that this kind of landscape is something to do with different type or this different possibility of the ground state, right, of your protein structure, right? Is that, am I correct here?
Yeah, it is, because there could be a lot of energy minima that can occur, and then we are not very lucky that, like in this case, even if it is schematic, but it also reflected some truth that that the native proteins that those are the protein that is fully function and useful, has higher energy than the protein that is dangerous.
Okay. Yeah.
So it's a matter of time that it could be perturbed and jump across the barriers and becomes the one that causes disease.
Okay. All right. And I think that this protein engineering or proteins physics is one of the branch. It's a branch of the, I would say, complex system, right?
I think so.
Right. So because you have like, well, you when you put all that molecule together, and then it emerges some very unique property out of that single molecule that you ensemble in them together, right? Something like that. I think this is going to do something to do with the complex system, but in a very complex way, I would say, right? Okay, so yeah.
It's just like our approaches are like we started with seeing the result first, and then trying to find some good way to explain, and then but even if we have some good results and which could in some way reproduce the experiment, but we still feel that we have a lot of like missing piece of information. So that is why we trying to jump more into treating this as a complex system problem.
I see. Okay, so next is something to do with the COVID-19. Is that this kind of protein engineering or protein simulation? Well, something that has something to do with studying structure of the COVID virus. I don't know. Is this something that we can use this kind of technique to study the property or the evolution of the COVID-19? Can we use this kind of technique to do that?
Actually, for COVID-19, there are a lot of target proteins to be studied, and this main protease is one of them. But if I try to draw a picture of if I try to draw a picture of a COVID cell, like this.
Okay.
It looks
okay. It looks like an alien now. Anyway, okay.
So now a lot of people they're trying to play with spike proteins.
Okay.
And antibody.
Okay.
Which are like our vaccines.
Okay, I see. Okay.
And then now we are playing with the main protease. So normally, if many people are like familiar with robot model like Gundam or something, so let me explain that normally when the COVID, coronavirus, they try to reproduce themselves. They're kind of like producing the proteins into package. So this is like protein number 1, glue with protein number 2, glue with like 3, number 4 in the same chain. Just like you have the robot model come at one piece at the first time and break it.
Right. I see.
Yeah. So main protease is the protein that like trying to cut between 1, 2, 3, and 4, and before they get like reassemble.
I see. Okay. Okay.
So that is why they call like the main protease.
I see.
Cutting the proteins.
Right. Okay. Okay. Well, very interesting. Okay. Another point is that So I don't know. Have you finished yet? Explain this coronavirus.
Okay. I think that is it. Yeah. So we also try to look at interaction between interface, which can be studied using MD simulations, because now in the PDB or in the protein data bank, where they give the data of the protein structures from experiment. So not from the AlphaFold. So AlphaFold take the data from PDB to learn. Yeah, but here the real data on the COVID-19 or proteins under COVID-19, there are a lot of them because now it has been in attention for the past few years. So we have a lot of like resource to play with.
Okay, very interesting. Next thing is about there is some you mentioned about there is some kind of living organism actually can be survived somehow in the harsh environment, like in the Yellowstone here. And I love the way you put the concept like a thug life. Okay, yeah. I can survive even in the harsh environment somehow.
So this one it's actually like my signature.
Indeed. Actually, I knew that. This is very interesting because it's somehow this kind of surviving living organism here is something to do with you mentioned later about mutation, right? Yeah, because it must be something special in it, right? Otherwise they cannot survive in this harsh environment, right?
Yeah, exactly. So this is actually somehow related to protein engineering, but it could be said that it's a protein engineering done by God.
Done by God.
Yeah, done by God.
Okay.
So it could be like they some point mutation in the DNA sequence also cause some point mutation in the protein. And then point mutation in the protein, such as what we did in the lab, it could just only one amino acid, but it could strengthen the proteins very significantly.
Yeah. And can stand the high temperature, right? Kind of very amazing.
Yeah, and then the pattern that see from the extremophile that if the protein like fold like this, and then they have some kind of cross-linking between the polymer chains made by disulfide bond. These are the patterns that we see in many of the enzymes in the extremophile. So this is how we see it evolve like this, and then we try to do the same in our enzymes to add this kind of cross-linking too.
I see. Okay. So next very nice question. So in the long future, is it possible to use this kind of study to like, I don't know whether is it going to be possible or not, but I just wonder that actually if we learn already a lot from this this living organism here in the harsh environment, can we kind of engineer ourselves somehow to stand the high temperature, and then we can live in that harsh environment?
Oh, very interesting.
Is it possible?
Okay, so let's think about this. Oh, that's very interesting questions. But we need to think about how many enzymes those are very like vital for important to life, and I think that we have quite a lot, and it might be impossible for site direct mutation. So my method here is site direct mutation to study it hard and then try to pinpoint the possibility and then it's designed to do the mutation. But so that one is impossible. But some impossible techniques are the directed evolution. So directed evolution is a technique that won the Nobel Prize in chemistry about 3 years ago. That it's just like they just kind of like adjust the environment to push the evolution to happen, to push like the probability for evolution to happen, and
to make it faster, right? You say.
Yeah. They kind of like filter out. They also do natural selection manually.
I see. Very interesting.
So that they can guide the mutation to occurs in right direction.
All right. Okay. Anyway, interesting. And then another interesting part that you mentioned, actually, with this kind of simulation, you can first predict what kind of structure that we can I don't know, we going to have, and then later we do it in the lab, right? I mean, I don't know which slide, but you at some point you mentioned that actually you do the simulation first, and then after that you can use that kind of simulation as a criteria. This picture as a criteria to make the in the lab somehow.
Yeah.
Then this going to be reduce lots of the cost behind the research, right? Because we are not going to randomly choose all kind of possible configuration of the protein, right? But exactly. with the simulation, it's going to guide us what is the right one that we need, right? Somehow.
So now so there are two sides of people working on this. So
okay.
In one religion, they believe in doing
religion. Okay. You have to. Okay.
They believe in doing random mutagenesis, and then they kind of purified, and then they do the sequencing to see which one that like best. So it kind of like high throughput technique.
Okay.
I think this is good, but if we're not rich, one thing.
Yeah. Okay. So I would say that the traditional way is like an orthodox thing, right? Like you are not the orthodox, right? Somehow you need we need a cheap budget, right, in order to do research, and this is would be the answer for that, I believe, right? Okay, so
we do the side
okay, so that at the very end is very interesting point, because you mentioned about network system, and I believe that something much to do with the topology as well, right? When you put this kind of graph, this map, and so I just wonder that you mentioned about the operator A, that you put the adjacent matrix here. And then when you consider A to the K, you say that actually you're walking the K step or K time, right? Something like that. And then this is something to do with like random walk.
It is, it could be thought as a walk on this topological network. Yeah, it's a walk on this grid. But it's not here. It's not like a random walk experiment. But we're trying to find a number of all possible states, and then we define it as communicability or centrality.
Oh, centrality.
Okay. Actually, it's not defined by me, but it's by Estrada and maybe some other people.
Well, yeah. Well, that is a student here at our institute working on this networking. That kind of stuff, and then I listen quite many times about this basic network stuff, and then about this adjacent matrix and all of this stuff. And then, well, I still don't really get it. But well, it's very interesting anyway.
It's quite new to me too. Yeah, now Top is working hard on this. There's a student of Dr. Thirasith, no, UA.
Right.
She also used this tool to observe the network between transfer entropy between cryptocurrencies.
That
So changing this adjacency matrix to be
I see.
matrix and cryptocurrencies.
Well,
And
something
somehow to measure the network.
Well, something to do with money is definitely interesting. Okay, let's see, and then well, we might have a best way to trade crypto, right?
Yeah,
with this the study, right?
I feel very lucky to co-supervise this project.
Okay, let's see. Okay, very interesting. Well, let me see whether there is question from the online, or I don't really see it, but yeah. But there is some only greeting word from people online, from the online people.
Oh.
Not really a question, I think. Anyway, so well, I would say with this very interesting talk anyway, and then I learned a lot of things about this protein structure, and then I can see that actually this is not limited to like, well, in the field of biology. Well, when physics came in, where we can do a lot of things, I believe, and then well, it turned out to be also you can use the maths like network and topology to study it, right? This is kind of, well, networking of the different knowledge that you can put together to study things, right?
Actually, this is driven by student. It's a student-driven idea. So,
right, okay.
the student he said, okay, he liked biology, but okay, he also liked math very much. So, like he asked me, like, could you design the project to be a little bit more mathematics than you do? Yeah.
Okay. I see a reason why you came in. Well, you come in this direction because this is to do with the more in math, definitely. Okay. One last question before we end the talk. So, well, I see you mentioned you put on your slide about quantum stuff. But I believe that what you have been working so far is still in the classical regime, right?
Yeah, it is.
And then the question when we need quantum, then.
Oh, okay. So it just for molecular dynamics, we just need information from first principle or quantum calculation at the very beginning, in order to get some constants, such as if we approximate covalent bond as a spring, we need spring constant. So we need to get the accurate spring constant from doing density functional theory or something like that. Yeah. So that's when we use quantum mechanical calculations, but at the very beginning. Yeah.
Okay.
And then after that, we transfer the information from quantum mechanical scales and then bring it into the set of classical parameters.
I see. But well, this means that at this when you finish your computation, there will be no quantum effect at all, right? From your result.
Yeah, for this.
Okay.
Just only like thermal fluctuation.
Okay, I see. All right. But at some situation, you need to put the quantum back, right?
Yeah.
But there must be some situation that we need quantum effect somehow to taking into account.
For example, that if we really want to monitor chemical reaction when it happens. But now I'm just trying to see the chemical reactions before and after it happens, because it. But there is sometimes that is unavoidable that we need to look at it while it's happening.
Okay.
And if we want to look at some other applications, such as in the field of materials, we might need to simulate conduction bands, valence bands, and we change the tool to density functional theory.
Okay, I see. Okay, anyway, so well, I would say I think we come to the end of the talk, and I would like to thank our speaker again, Dr. Thana Sutthibutpong. Okay, and thank to everyone who stayed with us about a lovely 1 hour and 15 minutes here. Well, okay. And before you go, please spend your time a bit on doing the survey form for us, which is actually the link already given in the comments of this on the Facebook Live and also the YouTube, I believe. And then see you again for this Colloquium number 7, but not in next month, but we will resume this Colloquium again in March, okay? Because next month in February we're going to have public lecture. Then we're going to postpone the Colloquium until the March. Okay, stay tuned then. Bye. Thank you very much.
Bye bye.